And Dave, you should start recording as well. Okay. So Ron, we need to hear from you, and
yes,
we'll be on our way.
Okay. Can everyone see the screen?
We can see the screen.
Oh, great. Well, there's the first step. Now, can you hear my voice? Since you just said yes, then you can. Fantastic. Well, good afternoon, everyone. I wish I could be there. It sounds like you're having a great time because I heard a lot of laughing. And it took you a long time to get back into the room. So it must be a fun meeting. So that's fantastic. It's important to have fun. Yes, it's what, a little after 4 here. It's not so bad. It's kind of a gray day, but it's warm. Yeah, just a quick about me. Not a lot, but I've been here at FDA a little over 3 years, but about 25 years or so in industry and then some consulting as well. So at Purdue Pharma in Stamford, Connecticut for about 16 years in biostatistics, data operations, and informatics. And then at Daiichi as head of biostatistics and data operations and informatics there as well for about 6 years. And then I decided, well, it might be fun to work at the FDA. So I've been here a little over 3 years. And most of what I've been involved in is policy and guidance around study data. I am in the Office of Strategic Programs, as it says there, in the work in the data standards area, but also dabble in other areas as well of electronic submissions, the electronic submissions gateway. I am chair of that board, which any of you that are in the room that have an interest in the gateway and how submissions are pushed through and what happens to them, well, we're doing a lot of work to update the process and the interface with sponsor companies, and also chair an operations committee of our data standards program board, as well as some other stuff. So with that, yeah, I'm going to go through the guidance, the two guidances and the technical conformance guide a bit at a high level, focus more on the policy end of it than on the specifics of what's in them, though there will be some bits and pieces on the timing mechanisms, because I know that for some that has been, there's been a lot of questions about that. The standard disclaimer, everything I say could be held against me, so if I say something that doesn't make sense, that's me, it's not the FDA. The FDA does no wrong, as we all know.
Ron, one thing real quick, your webinar window on the right side is partially blocking the view.
Oh.
So if you just push that little, yeah, there you go. Yeah, it's
good? All right,
yep.
Sorry, that's my double screen that was showing. Okay, so a quick path to electronic standardized study data. My career goes back, this is kind of like my career, started in the 80s, and where there were no standards. All the standards were at each individual company, and we all did it perfectly, and we all did it better than the other company. We all had our standard, internal standards. But in the 80s, FDA, this was really during the NDA rewrite of 314, CFR 314, where there was a guideline developed called the FDA Clinical and Statistical Sections Guideline. And that document talked about submitting, that sponsors should submit SAS data sets or ASCII flat files. So that was the beginning. And Steve Wilson, matter of fact, is the who's in biometrics, is the guy who was the lead author back in, I think this document was around 1987 or so. But then fast forward into the 90s, and we see that there's a CDISC comes on the scene in 97, and there's a collaboration with FDA, and our friend SAS Transport is supported by FDA. And then in the 2000s, there's a formal FDA support of CDISC-based submissions, and going to 2012, really an important year where the FIDASIA statute law was passed, which added the 745A little a to the FD&C Act, and it also reauthorized PDUFA, which was the 5th iteration, so it's PDUFA 5. And then in 2014, so in those intervening years of 2012 to 2014, we started to put our pen to paper, our fingers to keyboard, to draft these guidances, the 745A FIDASIA guidance and the e-study data guidance. There were several starts and stops of those guidances, mostly because we knew that the FIDASIA law was imminent, and so we went out on draft again with the binding guidance as we know now, that soon became final. And then the technical conformance guide, I'll talk a little bit about that in a bit. So I've been showing this slide as long as I've been at FDA. Colleagues in OBI, Office of Business Informatics, have provided this information to me. And you could see that there's certainly been an increase in the submissions that have at least one or more data sets that are in SDTM studies that are SDTM or resemble SDTM. And you see where we are in 2015, 71%. We certainly don't expect any fall back into the 60s, given that many companies now are on the bandwagon, for submitting standardized study data. But we still have a way to go, because this is not, as it says, it's one or more studies. It doesn't say talk about the submission, the full submission itself of clinical and non-clinical studies. I show this slide as the value proposition really within FDA, CDER, but certainly it's important for industry as well. And we look at predictability, and I have CDISC there. As I said before, for decades, the reviewers didn't know what they were going to get, right? Every company had their standard, and now we are moving more and more that what comes in the door, the reviewers know that it's going to be SDTM, and it makes it a lot easier for the review. Traceability, we talk about that a lot. The statisticians look at data from the study report all the way back. So go to the case report form. The statistician in the industry and the statisticians in the industry, we look at data from the CRF going towards the study report. So it's important that the reviewers can take those breadcrumbs, if you will, and find that path all the way back and trace a data point back to the case report form. Replication, again, data standards affords that if we have common variable terminology, it certainly makes it a lot easier for statisticians here to replicate a sponsor's analysis rather than start trying to understand what a sponsor is trying to say in their in the data. Pooling of data, not only pooling across studies within a submission, but internally here at FDA, there's also interest to pool studies in the future as we get to that point, to pool across submissions, right? So a lot of data will be sitting and is now being transferred into the CTR. I think Lilliam will probably talk a little bit about the JANIS project and the CTR, the Clinical Trials Repository. So standardized study data make it easier for that. And tools, JumpStart. Lilliam is going to talk a lot about the tools. Certainly the standards, standardized study data. We're able to leverage tools that can be used across review divisions and across the biometrics groups. So that's very important. And communication. So internally, if we have a terminology, standardized terminology, it's easier to talk within the FDA, but also it's also easier to talk with sponsor companies using terminology, the same terminology. And all of this has an impact on overall data quality, transparency of what we're doing, and eventually the interoperability of not only of the maintenance of standards, but also a connection with clinical, with healthcare, right? Certainly this is not just all about regulatory review. FDA also has a mission connecting to public health, so it's important that there is that connectivity to healthcare. And standards and bridging between CDISC standards and HL7 standards is important. And all of this translates hopefully into more efficiency and faster decision making. So going to the Act, FDASIA Act, that was passed in 2012, this was Section 1136 of that law, and that law added the 745A. So when you hear 745A, that's a new section within the FD&C Act. And as I said before, it authorized PDUFA 5. So FDASIA, I mean, this particular 1136 passage in that statute is very brief. It's only a couple of paragraphs long, but it clearly authorized the FDA to implement electronic submission requirements, right, by specifying a format, and it says in guidance. So when we start to talk about binding guidance, right, it's kind of an oxymoron. How can you have a guidance that's binding when for decades, you know, guidance is guidance. It's recommendations. You should do this, right? We would like you to do that. Well, because the statute says FDA will specify it in guidance, the folks here in the Office of Chief Counsel interpreted that, well, we can specify that those requirements in a binding guidance. So that's the birth of binding guidance. And I mentioned PDUFA 5, which now goes back to obviously 3 years, and we have a couple more years to go. But it specifies this is a handshake document. This is just a document between FDA and pharma that says that we will do, here's our commitment to do certain things with respect to, this is a pretty long document, but there's a section 12 that talks about electronic submissions and standardized study data. It says we will develop standardized clinical data terminology, right, i.e. using CDISC. Well, that, we interpreted that as being a focus on therapeutic area data standards, and I'll talk about that in a little bit. And it says FDA will publish final guidances specifying data standards, formats, and terminologies, and certainly we will be doing that, and that is through Federal Register notice. So what does this e-study data framework look like? So the little umbrella circle there, the FDASIA guidance, the 745A little a guidance, is that umbrella guidance. It basically says, okay, FDA will specify individual guidances that are content-based and will give you all the detail on that particular area. So it just sets the tone that FDA has the authority to require electronic submissions and specify the format of those submissions. The e-study guidance, as I said, is a guidance, binding guidance, which talks about the requirement for standards. And it focuses on the data standards catalog. A companion, and there could be more of these, but the ECTD guidance, which is Electronic Common Technical Document guidance, is an example of an individual guidance, binding guidance. Two key components there is the data standards catalog, where the supported and required data standards are located, and that is on our FDA website. And an important document, the Technical Conformance Guide, is a compilation of some prior documents and a lot more additional information on how to submit standardized study data. So how will all this be implemented? Well, I think most of the folks in the room know this by now, but just to go over it, we know that the final guidance published December 2014, the draft published in 2014 as well, and we got the final in the same calendar year. The clock has started, and so 24 months after the final guidance, these individual, the FDASIA guidance will be in an implementation phase. But as I said before, the implementation, the FDASIA guidance is just the umbrella guidance. It's the individual guidances that focus on, if I had a pointer, I'd be looking at that large white circle on the right-hand side, will be focused on NDAs, ANDAs, and BLAs and INDs, and will specify timetable, content, and format, right? And so now drilling down to the e-study guidance, when will standardized study data be required? Well, that published as well in December. It published December 17th, 2014. So the clock starts. So in December of 2016, standardized study data will be required, looking at the big circle with the big green check, but it's for studies that start must use the standards in the data standards catalog. So point here is that it's for studies that start after December 17th, not on December 17th. If you got a submission on December 18th, everything in your submission has to be compliant to the standards in the catalog. No, it's for studies that start. Very important point. The e-study, this is for some might be kind of difficult to grasp. But we try to be as consistent and transparent with when stuff is going to happen. So we fixed on a month and a day, March 15th. So when we issue a Federal Register notice surrounding study data standards, whether it's a new standard or a upgrade, update to an existing standard, we will use this March 15th transition date. So the transition date is the next calendar, March 15th, and I'll have an example of what this looks like. Following publication of the Federal Register notice. The transition date that is talked about in the guidance does not indicate the required date, but it is a component to get to the required date. All right, so we issue a Federal Register notice announcing a new standard, right, on September 5th, 2017. So because September 5th is after March 15 of 2017, we go to the next calendar, March 15th and year. So next March 15th, following Federal Register, is March 15, 2018. So the requirement date, because this is a new standard, and the guidance says that if FDA is announcing the support for a new standard, it will always be a 2-year period. So 2 years, 2018. So March 15, 2020 for studies that start. 2 years, so it's 2 years after, and studies that start after March 15, 2020, that new standard would be required to be used. So what are the data standards? I mentioned the standards catalog. The e-study guidance is standard agnostic. It gives examples of some standards, but it doesn't specify the standards. You have to go to the data standards catalog to see what standards are currently supported. And we just issued version 4.1, I think about maybe a week ago now, and you could see that there's a column, I don't know if you could see that in the room clearly, but the date requirement begins is now filled in. So you could see that clearly if you open up that Excel spreadsheet on your own. How study data standards will be required? Well, the technical conformance guide is a non-binding guidance. It is a technical, what we call technical specification document that is linked to the e-study data guidance. And as it says there, if you could see it, it's incorporated by reference into the e-study data guidance. We are now at version 2.1. That was posted last month, and it has a lot of information in it on how to submit standardized study data, things that we have concerns about, and so we tell you about those. It supersedes some older documents, the study data spec document, and the CDER Common Issues document. So you can use it right now. Some of the key points that are new, really new key points, in the planning and providing standardized study data section, there are 3 key documents that we are recommending that sponsors submit. One is the Study Data Standardization Plan for both clinical and non-clinical studies. Many companies are already submitting and have been doing so for a while, submitting the Study Data Reviewer's Guide, and we're asking, suggesting, recommending that you submit an Analysis Data Reviewer's Guide as well. And both of these documents, the SDRG and the ADRG, there are templates that have been prepared by industry volunteers via the PhUSE organization. So in Section 8 is a new section talking about data validation and traceability. Very important section on legacy data conversion to standardized study data. It doesn't tell you how to do it, but it tells you what we would like, some of the caveats, if you will, things to be aware of when converting to standardized study data. Are there waivers and exemptions? Well, there are no waivers from the requirement to submit data in conformance with the standards posted in the data standards catalog. You can have a request a waiver to use an older version of a standard, for instance, SDTM 311, which now is in retirement. That was retired in January of this year. If companies had studies in 311 and wanted to not up version that, they could request a waiver to submit 311. Are there exemptions from the requirement? Yes. There's exemptions for non-commercial. I won't go into detail. This is specified in the guidance for non-commercial INDs and for some BLAs. And can FDA refuse to file, or in the, with respect to ANDAs, refuse to receive? And the answer is yes. That was specified in the guidance, but however, there's certainly no desire for FDA to want to refuse to file. That's a review division responsibility to do that, and we feel strongly that if we want to look at a submission early, very early, before it gets to the review division to see its conformance to the standards that are posted in the catalog, and reject the submission, not have it move into a refuse to file situation. I don't know, interest of time, I don't know how much more time I have, but the clock has started. I mentioned that. We want sponsors to use what's in the catalog now. If there's multiple versions of a standard, certainly sponsors can use whatever they could pick, whatever one they want to use. Certainly we would recommend using the most recent, but if there's more than one version, they can select the one they want to use. Updating the catalog, we will update as needed. If we find errors, we will update it as we issue Federal Register notices, which we recently did for WHO Drug Dictionary, which was never in the catalog. That WHO DD is now posted in the catalog. We all know that for decades we have all coded ConMeds with WHO DD, but it just was never in the catalog. So we wanted to specify that, and we did that by Federal Register notice. I mentioned the transition date, always March 15th, and a requirement date is that it will always be 24 months for a new standard, and it's 12 months for a version update to a standard. And how does FDA determine a version update or a new standard can be supported? Well, we execute an internal process to see whether we can process, review, and archive a particular update or standard. And if we can, then that is, after we execute this process, we then will write the FR notice to inform the public that we can support the standard. Is there a limit to what can be required under FDASIA 745A? The answer is a qualified no. The qualification is that if it's it would have to be determined, it would have to be reviewed by our Office of Chief Counsel, but by and large, if the data or information is part of a submission under Section 505 BIJ of FD&C, it may be within scope of 745A and could be required by FDA and this format specified by FDA. And are the recommendations in the conformance guide required? No, but we certainly encourage sponsors to submit using those recommendations. Well, we're not really sure. At this point, we issued the second version of it, and then because there were some errors in the document, and we recently did 2.1. There's a focus on a version 2.2, or maybe it'll be a 3.0, not sure yet. So we're not really sure how often we're going to do this. We think that we need to see how this tracks. We meet internally as a multidisciplinary group to review any. Sometimes we get comments through the eData team that will inform us that perhaps we need to update the conformance guide. Let's see. Well, I issued that. So the last little bit I want to talk about is the therapeutic area standards. We've had an ongoing, because of PDUFA 5 commitment letter that I mentioned earlier, we have a process in place for past couple of years now to collaborate with CDISC and organizations that, like the CFAST group, which is I'll show you a slide on that, to ensure that there is public input into any development of standards. So very open, very transparent process is important to the FDA. It's important that any of these standards are sustainable. So if there's, for therapeutic areas, if there's data elements out there in a medical clinical society or available in NIH, because they house a tremendous amount of terminology in their NCI enterprise vocabulary system, we would adopt that or adapt it. It's important for sustainability. We know about CDISC Share environment, the metadata repository. We feel that that's an important system that will ensure sustainability and consistency across all of the CDISC foundational standards, and of course the therapeutic area standards, which are part of the SPTM structures. And of course, as I said, we'll be implementing in guidance or FR notice as needed. We have in our here we've focused looking at the upper left-hand piece of the pie. We focused on efficacy endpoints internally and produced some recommendation, informal recommendation documents to feed the CFAST initiative that eventually goes to CDISC for the development of these TA standards. And that CFAST initiative, I'm sure by now many of you know clearly about the CFAST initiative, Coalition for Accelerating Standards and Therapies, which is originally started out as a formal partnership of CDISC and the Critical Path Institute, and then became this structure, as you see on your screen, that is composed of a scientific advisory committee and a steering committee. And FDA, as you can see, sits on both of these structures. And it's we interact with them on a, let's see, I think bi-monthly basis in developing these standards. So we talk and work with the review division to provide input into these standards. And so quickly, the top section is the CFAST initiative, and you see FDA's role when CFAST is developing a particular standard, and we are not engaged in a recommendations paper internally at the FDA. We have input points at the scoping and planning stage, and internal review, and of course in the public review, to look at those standards to provide input to CDISC prior to publication of that standard. When we have developed formal efficacy endpoint recommendations, we've followed that process that you see below. So far, this is a slide that goes back to November, needs to be updated a bit, but you could see on the left, the red, the CDISC released therapeutic area standards, and you can go to their webpage to see any updates. And then in the blue bar, all of the TAs that are in various stages of progress, either planned or actually undergoing development. So a tremendous amount. And this little thermometer shows there's approximately 58 therapeutic area standards that we have targeted, and that's posted on our website. It kind of fluctuates because we've had reviews by the review divisions over the years. So started out as 55 and then going up to 58, kind of held around 58. But you see that we are certainly up in the mid 65% or so, having done some work on 65% of these therapeutic area standards with a couple of years to go. And there's various support. You guys probably know about the support. The FDA webpage, study data standards resources page has a lot of information on helping understand standardized study data and links to documents. The eData team fields questions from industry on, and we meet on a regular basis on data standards questions, and there's a complement E-sub team that focuses on mostly at this point ECTD questions, and Lilliam's Office of Computational Sciences, and our CDER Office of Strategic Programs and the Data Standards Program, and CDER's Office of Business Informatics collaborate daily to work on the data standards and leveraging them for a more efficient review. And with that, I'll say thank you.
Thanks, Rob. So we're going to pass a microphone around the room to people who have questions and people who are online. We've got about 75 people or so who are just at the web conference. If you want to sort of put your questions, enter your questions in the question box in the webinar, we have somebody who's going to be catching them on our end and passing them through. So we'll ask them. We'll ask them in person here at the podium. But let me start by turning on the wireless microphone. And I wrote down a couple of questions, which I'll start with while people are sort of thinking about their questions. And maybe you can pass the wireless around. Question in the back. So first of all, Ron, let me make sure that you can actually hear me.
Yes, indeed.
Okay, good. One of the questions I wrote down for myself as you were talking was you were talking about when a new standard is about how to determine when a new standard is going to be required for a study. And so when you're talking about new standard, are you talking about entirely new standards or new versions of standards? So for instance, would we think of version 3.1.3 and 3.2 implementation guides in SDTM as different versions or different standards?
Well, I would say that those are different versions.
Different versions, and so
yeah.
and so if a new standard becomes required, for instance, the Atom standard, which is not required yet, right? It's not specifically.
Well, no, Atom is part of the puzzle.
Okay.
So an example would be. So we just issued the Federal Register notice for WHO Drug Dictionary. And because it was never in the catalog, and we have considered it, but we know industry is using it now. But because it wasn't in the catalog initially, that would be considered a new standard, and that would be under the 2-year clock.
Gotcha. Okay. So if it's a new version of a standard, then it just goes into the catalog and it becomes available for use, but it's not a requirement.
No, if it's a new version. So SDTMIG 3.2, right? It's currently not in the catalog, and we will eventually be issuing a Federal Register notice that says FDA supports 3.2. It will get posted to the catalog. It will be considered a version update to SDTM, which means it will have a requirement date, but that requirement date will be a 1-year requirement date. So for studies that start 1 year after the
March 15th transition.
After the March 15th date will be required.
So 3.2 then would be a requirement, not just a choice.
Oh, right, absolutely. Yeah, absolutely. Right.
Okay. That was one of the things I was interested in, and a second thing was you were talking about the therapeutic area user guides at the end. Are those also going to be sort of in the catalog as requirements, or are those going to be sort of in the realm of, we'd like to have it, we'd like to have things using these data structures. Are they going to become official standards that get mentioned in the catalog?
Yes. Yes, they will be, we will do a Federal Register notice announcing the support of those as well.
Okay, okay. Do we have questions?
We have a question back here.
Hi, Ron. This is Tom Gitzer. Quick question. I think it's a clarification, hopefully. You mentioned the requirement date for clinical studies, and just a question is that many of us think of the pivotal phase 3s or a certain section of the application now that might use. I'm assuming that applies to all phases, so phase 1 through 3. I don't know if you would need to go as far as phase 4, but that's the question. What phase is the studies that
Right, yeah. Good question. That's a good question. Good question. We've had that question before, and we have not specified phase 1, phase 2, phase 3. So our feeling is that the data for phase 1, phase 2, phase 3 will have to be in compliance with the standards listed in the catalog.
Any more questions? Okay, we got another one. Couple more. Okay.
Okay. This question is coming in from online here, and because I've already seen the slides, I know the answers, but you can go ahead and answer. The first question is there a template created for the study data standardization plan?
Why? That's another good question. So to my knowledge right now, there's no template available. We do know that I'm getting some feedback, so that's why I have hesitation in my voice. Never liked the sound of it. But we do know that the PhUSE organization may have or does have an initiative underway to develop a template for the study data standardization plan like they did with SDRG and ADRG. What we have done internally here, we have developed a recommendations document, not a template, but we've taken what is in the technical conformance guide, that little brief section, and expanded that a bit with some additional recommendations, kind of just blown that up a bit to clarify what we meant in that section. And the plan right now is that we will issue that recommendation in a Federal Register notice so that folks can make comment to it. Because we've been getting a lot of questions on that, so that's a good question.
Yeah, and I would add to that that's actually one of the projects being worked on by in the CSS PhUSE working groups. It's actually the biggest project really that's being worked on, and we have top men in the room who are working on that, and actually we're going to hear about the progress of that later in this meeting.
Okay, there's another question here, comment question. I'm glad to see that 71% of all submissions are SDTM compliant in 2015, but how has it been received at the FDA? Has it made a difference in reviewing the SDTM data? We always hear that the SDTM is required, but never if they made a difference. We really haven't heard from actual reviewers that it was helpful and reduced the review time.
Okay, that may be a good question to re-ask after Lillian's done.
Right.
Is there anything that you want to say about that?
Well, I mean, yeah. So, there's still a lot of the data that's coming in. I think. Well, so let me preface my remarks. I know Lillian's going to have a lot of comments in her talk about leveraging standardized study data for the tools that her office has deployed. And so you'll see the difference that that has made. As far as metrics, certainly there's a lot of interest on the metrics of how much faster has standardized study data made the review. Honestly, I don't think we're there yet. I don't think there is enough data, if you will, to be able to make that assessment. I think if you went to specific review divisions, maybe you can do that, but as an agency, or even that would be very difficult, but as even in CDER, I think at this point in time, it's a bit premature to be able to say, has it made a difference on a CDER level? I'll just stop at that and let maybe Lilliam can address that in her talk a bit. But it's a good question, and we get that question from pharma with a capital P organization all the time, because there is a specification in the PDUFA 5 commitment letter that talks about metrics, but we're just not there yet.
A question from the back here.
Hi, Ron. This is Citron. You mentioned about the importance and usefulness of traceability of data back to the case report form. Can you speak a little bit about what you'd like to see from a reviewer's perspective of what you can do to help you with that traceability as it relates to ADAM or SDTM? That goes maybe beyond what the defined XML does now. If there's any personal preference that you have, insights, that would be great.
Yeah, the first part was a little softer. I just couldn't hear that first part. I heard, do you have any recommendations? Could you just give me, just reiterate a bit?
Sure, yeah, I tend to speak a little soft. It's mainly about traceability. What I was curious about when you mentioned going back, tracing back the data back to the case report form, and I was asking what you'd like to see going beyond the defined XML that we currently submit, how we can provide the traceability of data for you from a reviewer's perspective. If you can give some suggestions to clarify that.
Yeah, so a quick answer to that is there's as I mentioned, there's section 8 in the technical conformance guide that gives some understanding of what we mean by traceability, and so in the world of data standards, we know companies, many companies are submitting SDTM, but they're submitting SDTM at the end game, right? So they're converting their operational data to SDTM, but the analysis data may be disconnected or is disconnected from the SDTM. So you're not creating your analysis data sets from the SDTM. And so there's that path, that traceability is a bit broken, trying to go from analysis data set, non-standardized, to SDTM and back to case report form. So what we're recommending is that, and it's going to take a while, but we're recommending that SDTM, certainly you can go all the way back and start with C-based CRS, which would be optimal, right, to SDTM, and then from the SDTM data, create your analysis data sets. So that's the short answer, or maybe long.
I think we have time for one more question, and then since it's 5 o'clock for Lillian, we're going to need to move on.
Quick question, Dr. Fitzmartin. As far as the FDA is concerned, how many people do you have at the FDA right now with a computer science background that are looking at these standards? The reason being is I come from a computer science background, and I've looked at the SDTM. And from a computer science background, first of all, it's not efficient in storage, and it's not efficient in collection either. So the traceability, actually, the SDTM data sets introduce an error, a possible error in our submissions.
Well, so I can't, I have no idea about that. Maybe, again, not to toss this to Lillian, but maybe she has a better feel for that. But as far as the complement of folks with computer science background, there are a lot of people with statisticians and data managers and experience or background, but I don't know. I can't answer that question, so I'll have to defer that.
Okay, we'll take a rain check on that one.
Yeah.
Patty, you have one more?
Question?
One more, okay.
We'll sneak it in because Ron didn't have a question answer yet. Hey, Ron, balance between the nimbleness and the adherence to guidance. So I know that study data needs to adhere, of course, to our guidances, but when we have exploratory data coming in, it's a fine balance between biostatistics wanting to look at certain data and being afraid that the FDA will want to see same data, and there will be a rush to conform and to change the data at the last second. What are your thoughts on this? Do you have a logic as to what we should be prepared for to actually transform?
Well, so it's certainly the conversion of legacy data to CDISC to SDTM is a business decision. So if you want to up version or convert legacy, that's your decision now. But so we don't really make a recommendation on how to do it, but we certainly give suggestions on when you do it, what to take into account. When we get into the world where you have data that has not is not standardized and you need to submit it, you're going to have to submit it in conformance with SDTM. I mean, it won't be accepted, right? So now the world is yours. There's no requirement to receive converted, that you have to convert data. However, as you'll hear from Lilliam, a lot of the tools that are being deployed leverage SDTM, standardized study data. So I would say that once we get to the world of required data standards, anything that hasn't been converted will have to be converted. There's no two ways about it, because the binding guidance doesn't give exemptions, doesn't give exemptions for that, or exceptions.
Thanks, Ron. Okay, we're gonna stop the questions there, because Lillian is now,
Yeah.
I'm sure drumming her fingers going, when do I get to talk? So thank you again, Ron, and let's have that. And
did you take back presenter rights here?
We will do that.
Okay.
So Dave, are you going to be able to make that transfer of presenter rights to Lilliam? All right, and we see Lilliam's on the screen. Lilliam, can you hear us, and can we hear you?
I can hear you fine. Good afternoon, everybody. Can you hear me?
We can hear you, and so do you want me to give you a short introduction, or do you want to just give your own introduction? I was so good at Ron, so.
You know, sadly, I wasn't able to participate in that, so I would love it if you did mine.
Okay. Okay. Lilliam is the, she's been at the FDA since 2000. Prior to the, so she's now the director, yes, of the Office of Computational Science. And prior to that, she served as associate director in the Office of Science and Innovation, Office of the Chief Scientist, provided strategic leadership and support for innovation in FDA science. And she's also worked in the Office of New Drugs, leading development and implementation of the new labeling policy that included structured product labeling. So she's got a background in implementing standards at FDA. I stole this text from the PhUSE conference thing. So Lilliam, you might want to fill in a little bit more if you think some other part of your background is appropriate for this. And otherwise, I'm turning this over to you.
Well, thank you very much. And again, good afternoon, everybody. And thank you very much to Ron for sending the tough questions my way. I really appreciate that piece. But be that as it may, so what I'm hoping to do today is to tell you two major things. I want to tell you about some of our services, particularly JumpStart, which I understand there's a lot of interest in. But I also want to tell you some of the other initiatives that we are conducting in our office that are really driven to maximize the use of standardized data to help regulatory review and to help the decision making to be more effective and more efficient. So one of the nice things about actually doing a webinar is that you can change your slides very, very quickly. So what I decided to do was start with this and tell you that really what we are, I firmly believe that we are at a tipping point right now. We are at the tipping point of transforming the regulatory review because we have now the perfect storm for aspects that are going to help us get to the other side, in which we are going to be able to improve access to study data, to be able to really use that data to support regulatory decision making. And as you heard from Ron, the first piece and a fundamental one is the policy piece. With the final publication of the e-study guidance and the technical conformance guide, we really have a solid base for saying we are requiring the submission of electronic standardized data. And with that, we're saying we are going to leverage that data in that format to make it useful and a fundamental piece of the regulatory review process. And Ron is fantastic at taking us through the implications of that policy piece. The other piece is that, the reality of it is that we've been receiving standardized data for about 10 years now. The quality of it has been increasing, and I think that we are finally, both as a regulatory world and the industry, we're learning about the benefits and the drawbacks of the formats that we've chosen to accept. And as I think one of the questions about SDTM, and some of the limitations, I think we recognize that no standard is perfect, but we have committed to actually receiving the data in electronic standardized format. We have committed at this juncture to accept SDTM for clinical data, and for non-clinical data, and ADAM for analysis data. And I think that gives us a very solid beginning to really wrap our hands around how the data is submitted. And it brings in the third component, which is now we can use that data. We can take tools and technologies and draw services around that data and those tools to really provide, to end up providing reviewers with data at their fingertips, which is basically what my office is really interested in doing right now. So as John alluded to, I'm the director of the Office of Computational Science, and that is, it's a part of the Office of Translational Science in CDER. And our office, the OCS, is really the brainchild of Dr. Woodcock. About 5 years ago, she said, our CDER reviewers need a group that can really pay attention and address the computational science needs of the reviewers. And this is different from getting them email, getting them Word and, basic capabilities. This is a group that is going to try to help them in incorporating tools and technologies in the review process, and that's what we do. So we want to support, our mission really is to support CDER in improving that drug evaluation and review process, and we want to do that across the life cycle. Right? And we want to do that in a way that is really addressing how we understand our healthcare system to be changing. So how do we do it? So we do it by really supporting CDER in making decisions based on high quality data, based on the quantitative analysis of that data, and being able to affect the decision-making process for approving safe and effective products to be available to the public. So this is really what we're trying to do in our office. We live at the intersection of the data, the tools, and the technology. That's where the OCS really lives. And this is intended to depict that intersection, right? So we are driving to leverage the standardized data. We are implementing processes to ensure that the data is of high quality, and that includes data validation and other assessments. We're also working on initiatives that would allow us to integrate that data in a useful way by implementing a warehouse effort, which I'll talk a little bit about later on in this presentation, the JANUS Clinical Trials Repository. We're also working on making sure that we have the right analytical tools and the training so the reviewers can use those analytical tools. We also want to wrap those tools into services, and this is where I'm going to spend a lot of our time today talking about our JumpStart service. And at the end of the day, what we're doing is we're really impacting how the reviewers actually are able to work with the data. So this is what, again, I think that we are at a tipping point. I think that we can transform the way that we do regulatory review. Our office really is a service organization that is trying to leverage these different factors to be able to, at the end of the day, get useful data to the reviewers so they can be more effective and more efficient in their decision making. So this is very quickly to tell you how we operate. So what we decided is that we're, as I said, we're a service organization, and we drive on providing data and analysis support services. We also, and that includes, data validation, like I said, quality assessment, supporting the standardization, script development, direct support to the reviewers. We also work with tools and technologies. We want to offer the best analytical tools for the work that our reviewers do. We also want to wrap those analytical tools into services that are very directed to how we do regulatory review. And you'll get a better sense of that when I describe what JumpStart does. And then, we think that having all those things is really great. But if you're not really offering training and direct customer support, you lose a lot of opportunities. So we also spend a lot of time in training and direct customer service activities in our office. So I think this was one of the things that Ron alluded to as well, is that, high quality data is really the key to enabling regulatory reviewers. And what we are trying to do is offer up the tools, technologies, and services that are really going to help leverage that data and support decision making. So we feel that data standardization efforts are really fundamental to the way that we can truly use the data available to us. So this is sort of the vision that we have. Well, this is the vision that we have. We want to be able to have interactions with sponsors, right? We want to be able to be very transparent, very clear about what we need, and have that that two-way conversation between the sponsors and us as the regulatory agency. So that way the sponsors can submit the standardized data in a way that is, meets their needs. It also meets our needs for supporting the regulatory review. We want to be able to integrate that data, this is where our warehousing effort comes into place. We want to be able to access that data with tools and technologies, and we want to wrap it up in services and as is depicted in here for JumpStart and other analytical tools. So, so here's what JumpStart is all about. It's about applying the right tools at the right time for the right audience. And so I encourage you to go take a look at this whenever you have a chance. This link takes you to our external website, and it has a nice information that was put together when we submitted for an HHS Innovate Award last year, and we actually were awarded one of the secretary's pick for this service. So we're very proud of it, and it really helped us to promote it and to make it better in the last year. So this is what JumpStart is all about. JumpStart is a way of targeting a very particular time point in our regulatory process, and that's filing. So when an application is submitted, there's a 45-day filing period. And at that point, the review team needs to be able to make a decision whether the application in front of them meets the requirements for being considered filed. So the actual review process starts. Now, there's multiple pieces to this application, one of them being the data actually submitted for the clinical trials. So that's where we come in, right? So we want to be able to provide feedback to the reviewers about the data quality and also help them with exploratory analyses to support that filing decision. So the first thing that a reviewer does is they can submit for our service. So the service really leverages, again, the standardized data. So the first thing that they need to have is standardized data. They need to have a CTM. And then we make an evaluation, and we accept the application for review. So there's two components to the JumpStart service, and again, this happens all in less than in 30 days. So there's two components. One is the data fitness component, and then the other one is the exploratory analysis component. So the data fitness component is really to assist the reviewers in determining whether the data in front of them is really fit for review, and figure out the impact of any deficiencies that they find to the reviewability of that application. And now you have to think about the fact that this is the very first time that a reviewer will open up these data sets and be able to make decisions about the data quality. You also have to understand that we are talking about clinical and medical reviewers, right, that are expected to make a decision about the quality of standardized data within 2 weeks to make a decision about how that is going to impact the reviewability for the next 9 months. So, so what we do is within 2 weeks, we run a series of, validation activities, and we also do a lot of human exploration of the data. And within 2 weeks, we meet with them and we give them an assessment of what we found. And then based on that assessment, they make a decision as to whether the application is of high quality, or is of the data within that application is of quality enough to support the review. So then the other piece that we do is like 2 weeks after that, we do the exploratory analysis session. And the purpose of this session is to improve the efficiency of the review. So we do two major things. One of them is we set up the tools for them, and we also provide them common analyses. So not only do they have a set of basic analysis at their disposal, which we discussed with them as well, but also if they want to work in JReview, if they want to work in JUMP, if they want to work in NAID, which is our adverse events tool, all that data is set up for them to continue doing the work in there, so they don't have to worry about that piece. And that happens within 4 weeks of acceptance of the application to our service. So, so what happens is that in a month, basically, the reviewer has a very solid understanding of the data that is in front of them. They have an assessment of data quality, or what we call data fitness. They also have exploratory analysis that gives them an idea of possible safety signals that they might want to further explore. So it doesn't really end there. So we also do follow-up activities after a decision. And again, we don't make the filing decision. The reviewers and the divisions make the filing decisions. You know, after that, but what usually happens is that they're very happy with what they see, and they always come like, well, can you help me do this other thing, right? Or I want to set up the analysis this other way. So we actually have some follow-up activities, and we all as a closeout as well, we provide references to all the documentation that we provide for the output tables, and if there's any communications that need to go back to the sponsor, and we help them draft this communication so there's clarity about what is being asked. So that's part of our support activities. So again, the JumpStart service is an overall assessment of the data quality of the application in support of filing activities. It also provides the reviewers with exploratory analysis to really jumpstart their review process and particularly their safety evaluation of those applications. So going back to the data, so SCTM really is what enables that JumpStart to quickly analyze the data submission. So we, having the electronic standardized data, the tabulation data in this case, really helps us with the efficient review. It also allows us to create those standard analyses, which are the predictable basic analysis that we can provide to the reviewers. Things like demographics and AE tables, disposition tables, so they can get a sense of what's happening in that application, given that this is the first time they actually are seeing the data. So, so what I want to do in the next 20 minutes or so is I want to take you through exactly how we do it. And of course, I'm not going to be able to go in as much detail as they go in the actual JumpStart sessions, but just so you get a flavor of how this runs. So this is the first portion, and this will happen within 2 weeks. And again, this piece is about assessing whether the data is fit for review. And this is based on quality, it's based on the consistency of the data, and the analysis capability. So what you're asking really is the data of quality, and can I use it in the way that I want to use it to support my review activities, okay? And one of the things that we found out is the impact that this approach is having, and I just wanted to sort of point out some of the feedback that we've received in terms of how the reviewers really are responding to the service. At the PhUSE Computational Science Symposium, we actually had some medical officers come and speak about their experiences, and what we find in general is that they are really helpful. They're really happy with what they get out of the service, particularly because the thing that always comes back to us is, I feel in my level of comfort with this application, by filing as I would have felt 5 months into the review process, right? And that is tremendous. That is tremendous, in terms of how it can be more effective and a more effective review. Given that level of comfort of understanding the places where the data is really strong and the places where it might not be, and trying to get to resolution of those limitations. So basically, this is how it goes. So there's a data fitness finding discussion flow, and they cover each one of these activities. So again, I'm going to give you a flavor of how those discussions go and give you some examples, but we won't be able to go into the details. So they actually go through the data for each one of these pieces. So, so for the data fitness portion, they look at the study data reviewers guide and the defined file. They also look at all supplemental information, control terminology, and then they go through the different domains. And they look at the disposition information on net reconciliation. They look at places where there may be missing data, where there's maybe duplicates. There's also an in-depth look at standard units and race and ethnicity. The other pieces that happen is the term matching, which really allows us to look into places in where coding might not be optimal. The determination of the safety populations is also really important. And overall, these these assessments, by the time you have them all together, really give you a good sense about the quality of that data and whether it's going to meet the need of the reviewers. Because at the end of the day, that's really what we care about. So what the team does is they present the findings using this form, which is there's a question. They go through the findings and they point out the questions that you might have about that finding, and then they will outline the data fitness finding and how that might impact the analysis and ultimately the review. And this will make sense in a minute. So far, this number has changed, but so we're about 15 submissions now. The statistics for this particular presentation are going to be based on 10. We've done an additional 30 on FY14, and we started with a pilot in 2013 in which this is where we developed the service. So what you'll see here is we'll do the review question, the impact, and then I will tell you in the cases in which it's applicable, the statistics for what we're seeing right now. I think what's really important about that is that you'll see where people continue to make the same mistakes. And what I would encourage you to do is think about those places in which we need to really address whether the standard needs to address, whether there's need for clarity in the way that the standard is implemented. We also need to make sure that we understand where implementers need to make sure that they're doing the right thing, and where we may need simply more clarity. Because what we want to get to is a place in which the data comes in with less mistakes, so we don't have to do this back and forth all the time. So one of the first things that we look at is the study data reviewers guide, and then the review of the defined file. And this drives what our expectations for what the data is all about. So questions that may be asked is, were the SCTM data sets used as sources for the analysis data sets? It speaks to that traceability piece. If there are issues with the data, why did the sponsor choose not to fix them? It's a place where you can find information about the version of MEDRA or LUDRUX that the sponsor decided to use. It also tells you where things are located, right? Where are the efficacy files? Where's the safety information that if you're looking for a particular endpoint? And that's where the study data reviewers guide. And in the context of the defined file, how does the information in the annotated CRF map to the data set? And if the sponsor is using, do they provide explanations for the code to use in the data? So these are the questions that can be asked. Also, with the supplemental information, we would be asking questions about, what is the information that was included in supplemental domains? We've seen everything from really useful information to things that make no sense, that people didn't know where to put and they decided to put it in the supplemental domain. How does the information contained in these domains map to the parent domain? Can we establish the right relationship between the parent and the supplemental domain? And then what information from the supplemental domains could be useful for analysis? So again, this looks very reasonable and very simple to do, but when you've never seen this data in front of you, it's tremendously useful to start with these pieces and say, what do I have in front of me? What do I know about this data, and how do I move forward in the process of determining whether it's going to meet my needs? So in 2015 so far, and for every application that goes through JumpStart, the team will look at these pieces, the SDRG, the define file, and the supplemental domains, and then to be able to very quickly tell the reviewers, what are the relationships between the information that they have in the parent domains with the supplemental domains, and to highlight useful information, as well as uncover inconsistencies. And again, I want to make sure you understand why this is important, and the reason is because reviewers really, they're medical officers, they're clinical reviewers, and they, I firmly believe that they should be spending their time really thinking about the clinical implications of the data, and not just really doing Easter hunt, egg hunts of what, where is the data, where do I need to look for it, how do I get to it? I mean, we should be able to guide them so they spend their time thinking about what the data means and not about how to get to it. So control terminology is another piece, right? How does the sponsor terminology map to the control terminology? Which domains do not use control terminology? And this is really important. In cases in which the sponsor decided not to use control terminology, is that really causing ambiguity in the data? And that is one of the main reasons for spending a lot of time trying to figure out what something means. And I'm going to give you an example that 60% of the applications we have seen so far have issues with control terminology, right? This is something that can be easily addressed, in my opinion. So here's an example. Okay, so adverse event action taken, in this case, is not populated with control terminology. So the sponsor decided that they're going to write what they're going to write. So when you look at it, you say, well, there's palpitations and nausea, right? And they say dose alter and withheld. Well, I don't know what happened to that patient, right? That is not informative. And then that means that I have to go and figure out what happened, in reality, what happened to that patient. So the best way to increase the usefulness of this information is to say, here are these adverse events, and I am going to code them with the right control terminology. Now I know what happened to the patient. Was the dose increased or decreased? Did they decide not to do anything about it, so it was not changed? Did they interrupt it? Did they, withdrew it? So this is information that actually tells you what happened to the dose for that patient. It's a lot more informative, and it's more useful. So this is a good example in which control terminology really makes a big difference in the way that we can actually use the data. So this is one of my favorite ones, right? Because it's a really important question, and it needs to be asked every time you look at a review. And we always spend an inordinate amount of time trying to answer this question because the data is all over the place. So what do you want to know? Well, you want to know how many deaths occur during the study. You want to know where that death information is located, and was that information represented consistently across the domains. As it happens, there's 4 different places in which you can have death information right now according to the standard. And it's sometimes portrayed differently in each one of those places. So you end up with information that is represented in one place and not the other. So we have to spend a lot of time trying to figure out how that information matches and what is the reality of those counts. And this is a very fundamental question that we need to ask in every clinical trial. So I'll give you an example. So at the moment for JumpStart, this is one of the really, really useful pieces, is we do a death reconciliation in which 100% of the studies that go through their service, we provide a summary of this information, and it's how the death was represented in every place in which it could be represented in the standard. In terms of the death types, are they consistent with the demographics? Are they consistent with the adverse events domains? And even in the comment domain, where we sometimes find information about death, when there's that information is not represented anywhere else. So here's an example, right? And so this is an example where you have the death represented. You have a death flag, right? It says, okay, fine, there's a death here, and these are the reasons, and that's all good, right? So you have it, and you have a flag, and you have it coded in the disposition domain. You also have some information about it as a fatal AE, and you have information, and there's no information in the death domain, in the comments domain. Now this is another example in which now you have it only, you have an inconsistency in how it was represented as an adverse event for that cardiac arrest, and then you don't really know what happened between, you know, the third and the fifth, in terms of, you know, was it that the patient had a cardiac arrest and then was in the ICU and then passed away on the fifth, or is there an inconsistency on the way that the data was reported? So it just makes you wonder what the reality of that disposition variable is. The third line here, it's only, the information is really not even included. There's not a flag. There's not a death date. There's not a disposition. You know, the disposition code is adverse event. So was it that there was no death, or what is the reality of this line? And then in the fourth line here is an example in which, you know, for this particular subject, there is really no information about death unless you go to the comments. So how do you reconcile, how do you end up counting how many deaths you have in a clinical trial when the data is all over the place? And so what we do is we put all these pieces together, the demographics, adverse events, disposition, and comments domain. We put them side by side, we reconcile them so we have counts that are matched. And we know where the inconsistencies are. We know where we can't, sometimes we don't solve all the mysteries, but what we can say to the reviewer is, there seems to be some inconsistencies here. You have to make sure that that's not going to impact your ability to review or the decisions that you make based on this data. So I think it's tremendously useful to the reviewers. So another piece is missing data. You know, so again, you're receiving an application. It has data. You're not exactly sure what, none of you know what's missing. You know, and most importantly, out of what's missing, is it really going to impact your analysis and your ability to make decisions? Or are there any other variables that can be used in lieu of that missing data? So 60% of our applications are missing the seriousness criteria, for example, and have inconsistencies between the adverse event serious flag and the seriousness criteria. And this is just an example to show you, again, the first line demonstrating one in which you have a flag that says that there's no seriousness criteria, and that corresponds with the you know, whether there was a disability or a death or hospitalization or life-threatening you know, event. Line 3 also shows the correct way to portray, which is yes, there is an AE seriousness event criteria met, and then there's a reason why it was met. Well, the patient was probably hospitalized and had a life-threatening you know, event. Then lines 2, 4, and 5 show ways in which, you know, in number 2, they say there's a flag as a serious AE, but there's no description of about what is the criteria that was met for that to be a serious event. And then line 4 does the opposite, says there's no flag, but then they say, you know, there was something that happened. And then you have to go and figure out what that was. And the last one, you really have no idea, right? Because there's just not enough information. So this is just to show you that missing data can have an impact in the way that you actually end up looking at what you have in front of you and with the kind of decisions that you make on that data. So I'm going to go quickly through the next couple of things because I want to give you time for questions. But again, we look at basic, also basic sort of questions about, for example, does the demographics domain include start and end dates, right? Are the domains missing the epoch variable? Does the sponsor include the trial summary domain? Does the AE domain include a treatment emerging flag? So these are all things that impact the way in which you can look at the data. For example, the epoch variable was missing from key domains in 90% of our applications. Well, if you don't have the epoch variable, that really impacts the way in which you can analyze the timing of the events throughout the clinical trial. So it's really important information to have. The trial summary domain is missing in 30% of our applications, where this is going to be even more important as we move into our warehousing efforts, because the TS domain is the one that carries all that information about the clinical trial. So, it's going to be really important to make sure that the clinical trials are correctly represented in the trial summary domain. We all understand the repercussions of having duplicates. Continues to be an issue. Do any of the data sets contain potential duplicate records, and how does that impact your analysis? 40% of our applications are coming with duplicates. I think this is something that is very solvable, and it speaks to being able to reconcile the data. Here's an example in which the EG domains and the lab domain have records that have the same patient with the same test having different results. In the case of the EG, the ECG interpretation, you have one that is normal, one that is abnormal. It's the same. Which one is it? We don't know. The bottom one is the lab domain, in which, in one it says it's a fasting test, and the other one it says it's not. And depending on whether it's fasting or not fasting, those results may be normal or abnormal. So which one is it? We don't know. It's the same test. So term matching is actually one of the tests that are really helpful to understand how the quality, the coding pieces really affect what you do. And what we do is basically say, okay, when you're coding the data, are you really coding to what it needs to be coded at? For example, and I'll use the, I'll tell you also that 40% of our applications come with coding issues. So this is an example in which it's important information, right? And the information is missing because sponsors are coding things to other. And I've seen desk coded to other, which is a little disturbing. So, if you are coding things to other and they are relevant, then they're not counted. And this is really important to know. So what we do is we provide the reviewers with a term matching report that lets them decide whether the coding is good enough and where the discrepancies is, so they can make decisions about it. For example, in this case, subject withdrew due to worsening of pain was coded to other. Well, if this is a pain medication, it may be that it's not effective, so we need to understand why that decision was made and whether it makes a difference in terms of the decision for safety and efficacy of the product. A lesion to continue to grow, maybe that tumor drug didn't work. So coding to other really makes an impact as to whether there's an impact to efficacy. And then the third one is subject disease. Well, that's a test. That should not be other. That should be a test. So we can actually count that the right way. So standard units is a simple one. And do they use the standard units? What types they use? 30% of our applications are coming in with inconsistent units, and the examples are pretty straightforward. We spend a fair amount of time looking into safety populations and making sure that we are getting the right information, pulled together so the reviewers can actually make assessments that are, valid. And so that's one of the other pieces that we do. And I'm going to go quickly through these because I want to, again, I want to be able to give you time for questions. Race and ethnicity. This is a big one, right? 80% of our applications have had race ethnicity issues, and that's how is the sponsor actually indicating race and ethnicity? Where is that detailed information about the race and ethnicity located? And are there subjects that are missing race or ethnicity data? So that all is the pieces that look at the data fitness portion, and as you can see, it's a fairly comprehensive assessment that crosses all aspects of the data. We also provide, in the second portion of the JumpStart service, we also provide exploratory analyses. And that's to give them, it's to set up the tools to run the common analyses and point them out to signals that they might be seeing. And again, this is tremendously useful. They actually can take, we provide all of these analyses to the reviewers so they can actually continue working with them. This is to tell you that we run a variety of adverse event reporting analyses, and we pull them all together to really provide the reviewers with a good sense of what are the signals that are popping up. We don't tell them the signals. We just say, well, these are the things that are popping up. It is their responsibility to now, during the review process, to understand whether they're relevant to the safety and efficacy of the particular product. So we also have a variety of scripts and analyses that help them create to understand the lab data, to represent it, and to understand when there might be things that are affecting, again, the safety and efficacy profile of that product. So we want to do this very efficiently, right? So what we've done is we've taken the clinical reviewer template, which is what our reviewers use to guide the review process, and we've matched up these analyses to the clinical reviewer template, to certain pieces of the safety analysis. And what you're seeing here is the clinical review sections and how they match up to the analysis that we provide. Of course, we don't do it all, because the idea here is that we just want to point them in the right direction, and they will continue exploring those signals as the review process goes. So the last piece that I wanted to tie in is, so we have the data, we have the analysis, we have the tools and the services to help our reviewers. We're also working on a way to be able to warehouse the data and be able to provide it to the reviewers in a very useful way. And this is to show you the conceptual overview of the clinical trials repository, which is basically a data, well, it's a data warehouse with the ability to create these data marts that allows us to then connect tools and technologies to those databases and have the data have a layer of analytics and analytical tools and other sources of information coming into to help our reviewer make decisions about safety and efficacy of products. So again, I told you about what our vision is, and this is our vision for JANUS, for the JANUS CTR, again, to have those interactions, to be able to put them all together in one place and offer them to the reviewers. So what we've done is we've taken the CTR warehousing initiatives and we paired it up with JumpStart. So what we want to do is be able to offer the same analyses that we are offering with the JumpStart service and start to create a place where the reviewers can start doing this by themselves, right? So now you have a self-service piece to it. So we teach them how to do it, and it's kind of teach them how to fish. So we're working on ways to present the data to them in an integrated fashion, and then give them the layer of analytics that will help them explore the data, give them the layer of tools, more analytical tools that are more powerful and can do further analyses, and then guide them through that so they can be so much more efficient. So we're hoping that, so we'll have the CTR in technical, we have the CTR in technical production this year, but we're hoping to deploy it to all our JumpStart users next year, and we'll be able to see, we'll be able to continue developing the way in which our reviewers are interacting with the data and developing further capabilities for them. So as I told you at the very beginning, I do believe that we're at a tipping point. We really have the policy, we have the standardized data, and we're really working on those tools and technologies. And we think that that will put together the right environment for us to really transform the regulatory review process. And so again, I just want to leave you with this is the road, right? We want to have transparency. We want to have communications with the sponsors. We want to be able to leverage the standardized study data. We want to have the right infrastructure to integrate and to provide that data to the reviewers. We want to have the tools and technologies to really put that data at their fingertips. And we want to be able to put it in a way that is integrated and service-oriented. So at the end of the day, we have our reviewers thinking about the data, not dealing with it, and being able to make decisions that are contributing to more safe and efficacious products out in the market. Thank you for your time, and I will take any questions.
Okie dokie. I'll bet there's some questions for that. Okay, hang on a second.
Hi, Lilliam. This is Sandra Video from the Center. I have my Adam hat on, as I typically do. And I'm just curious as to whether you're considering using any of the Adam data sets in your tools. I can see where things like population flags that are ADSL, the treatment emergent flag in the APAE would be very useful for this type of analysis. And if you are interested, the Adam is very happy to work with you.
Absolutely. Absolutely. So I'm glad you brought that up. So, like anything else, I am a firm believer that if you try to do it all at the same time, you end up doing nothing. So as an office and as a service provider, we've been very we've tried to be really disciplined about trying to be really good at one thing before we move on to the next one. That's not to say that we don't want to use the Adam datasets. I think there's a lot of value in doing that. And actually, one of the things that I didn't mention that we do is we answer questions about the traceability between the SCTM and the Adam files when we have those available to us. So we do want to move in that direction. We are working on ways, and we work very closely with our statisticians as well. We're also exploring ways in which we can both do a similar approach to the Adam files as we do for the SCTM in terms of data fitness. We want to continue ways in which we can get better at establishing the traceability between the SCTM and the Adam. And then we also want to figure out what is the best way to incorporate in the warehouse our findings for both the SCTM and the analysis file. So we are thinking about it, and we want to pursue that direction. It's just a matter of timing. So I appreciate the offer, and I will certainly reach out to you.
Thank you, Dr. Lilliam, for the presentation on Genentech. I'm just curious, you have JANDIS, the clinical trial repository, and the JumpStart. In terms of the ordering, do you run JumpStart first before you load the data into JANDIS, or is it totally independent process?
I apologize, but it's really breaking up. Can somebody rephrase the question, please?
Yeah, so the question is, when you get the data, do you load it into JANDIS first and then run the JumpStart stuff, or are they really independent of each other?
Right now, they're independent. So we do, because we've been developing JumpStart, we've been in the development and deployment phase for CTR. So the loading up to last February wasn't happening at the same time. So JumpStart runs on its own schedule because we have a lot of applications and we have this tool set up to do the analysis. But what we're doing right now is we're loading up into CTR and exploring the ways in which we're going to provide the same service or as much as we can out of the CTR, in the future. So right now they're separate, but our intent is to be able to leverage the actually starting in October, the hope is that we will be, our loading process will be optimized, so we'll be able to load concurrently, and actually the analysis pieces will happen out of the CTR, like you're saying, which is what needs to happen, directly out of the CTR, so leveraging that. But right now, that's not the case.
Thank you.
Hi, this is Jennifer from Genentech. On the FDA point of view, the reviewer point of view, for the data cut, do you suggest to doing the DAS analysis, or you will suggest to do it in the SDTM, sorry. And another question is,
actually, let's pause there for a sec. Lilliam, did you catch that question?
No, I'm sorry.
Okay. So the first question is, when you have an analysis that uses a data cut, would you prefer to have that data cut in SDTM, or have all of the data in SDTM and the data cut be made in Atom? That's the first question.
I don't think, it really depends, and I'm not going to answer that because I don't know that there is a question. There's an answer. There's a 100% answer. And I think from our perspective, what we want to do is just to support the reviewer in understanding whether what they have in front of them is what they're going to be able to use. And in the cases in which we found out that the data was not cut in the way that was useful to the reviewer, or there was parts of it missing, or there were parts that needed to be recoded, or whatever that was, the really useful piece to this is that now you're going back to the sponsor during the 45 days of filing, not 3 months down the line. So we've actually, in actuality, we've moved that to very early in the review process, and now we can fix those problems, right? So if there's a need for cutting the data in a different way, if there's a need for presenting the analysis data sets in a particular way, now we're moving those discussions up in the review clock, right, to the very beginning, rather than 5-6 months on the line.
That's a good point. And then you had a follow-up question for that?
Yes. The second question is also related to the data cut. For the data cut, I think there's several of the methods was using. You can cut it in the event date. You can also cut it in the data entered to the database. However, if we do the submission, I think most of the time we do the event date, cut out the event date. But I just say, if there is any suggestion on the FDA, what you are looking for? Will you? Yeah.
Okay.
Go ahead, John.
So let me rephrase that a little, see if I got it right. There's a couple of methods that people use to do data cuts. One is data entry date, and another is a population definition of some sort. And the question was, is there a preference? And I suppose this is a question you'd really have to ask the review divisions.
Right.
Is there a preference for a methodology for data cuts?
No, and again, so that's not what we do. So again, that will be a conversation with the review team, and particularly with the statisticians. At this juncture, what we are really trying to do is just support them in making sure that within those first 45 days of filing, that they have the information that they need in front of them to make that decision on whether this data is going to be useful. So these are conversations that if there was a question about it, there needs to be a dialogue between the sponsor and the review division.
So actually, I have a question for you. One of the earlier slides, you had a little box down there.
Uh-oh, I can't hear you anymore.
Let's see.
Okay, now I can hear you.
Can you? Yeah. Why don't you turn off the microphone that's being passed around for the moment because that's causing feedback. Okay? A little better now?
Yeah, thank you.
Okay. So my question was, a quote from a reviewer, JumpStart said that the data sets were bad, so the reviewer asked for data sets that they could make use of in their review tools. And I looked at that and went, does that mean that what they got was not SDTM and what they asked for was SDTM?
And one of the challenges that we see is that because we've been doing this for a while, people have been submitting SDTM. And if you remember that chart that I showed you, I mean, I think it's over 60% of the applications are coming with the SDTM formatted data. I think the challenge is that it a lot of times is kind of like SDTM data. And what we're trying, and this is what really JumpStart shows, is that we can be doing better, and we should be doing better in the way that we're formatting this data to the standard. And then there may be places in the standard where we need more clarity and more consistency in the way that the data is represented. So I think that is it's about our reviewers really wanting to leverage the electronic standardized data with the tools that they have available to them, because what they really care at the end of the day is to make decisions that ensure safety and efficacy of the product, right? So, so I think I think the important piece about what we're doing is that we are really trying to move this conversation to earlier in the review process, because this is a conversation that would have happened anyway. It would just happen 4 months down the line, right, or 5 months, and then the sponsor will be like, oh my gosh, I have to go and redo this or send this with this other thing. Now we're moving them to filing, right? So that means that the reviewer can really start with data that is useful to them. So I think that it's just really taking advantage of being able to have these conversations so we can support each other in this decision-making process.
We have some questions from the folks online here. Okay, the first question is JumpStart available for sponsors?
No, JumpStart is an internal service.
Good try.
Right.
And the next question is, why is EPIC a must in AE and CM?
I'm sorry, why is what?
Why is the EPIC variable a must in AE and CM?
I'm not going to get into those discussions only because I'm not the person to ask the details about the epoch. My understanding is that the epoch really gives you time, bucketed information for what things happen during the clinical trial. And I think that the point of that is to say that when that information is not provided, you don't know when things are happening during the clinical trial. And that is an important consideration when you're deciding where the adverse events happen. But I cannot speak to why things are where in the standard.
Okie dokie.
Yeah, a question concerning the actual CRF data, what we call raw data. That data gets transformed into the SDTM data. Is there anything at the FDA where they're going to be looking at making sure that that information is being properly transferred from the original clinical database to the SDTM database? Because the structure in a lot of these domains are transpositions of what the natural collection of data is. And to me, it's very important that we don't miss data points when we submit to the FDA. But I'm wondering if there's any tools that the FDA is going to be using to look at the raw data to make sure that the SDTM is proper. Because to me, the SDTM data sets themselves are a separate, secondary, pre-atom processor.
So that's a really interesting and important point. I think if you look at the way that we've done review historically, we've never received the raw data, right? So you get the clinical trial, the summary, the clinical trial report, and then you get the tabulations. And then we moved into getting those tabulations in electronic standardized format. The way we've historically gotten to the raw data or the data capture in the forms is by inspections and by spot checks. So I do agree with you that this is an important component of it, and I think that we will get to a place in which we can establish the relationships of the data collection, the relationship of the data collection to the representation of that collected data into the tabulations, and their further analysis and further representation into the analysis files. At the moment, there's not a straightforward way to do it. It's an important component, but we don't really have a straightforward way to do it. What we do in JumpStart, though, is one of the things that the JumpStart team does is they check how the data sets match up with the clinical trial report and with the tabulations that were represented in that clinical. So we know that things match to each other, and that the data sets are in essence of quality, and that they match with each other. So that's at the extent that we're doing it right now. Did that help?
Yeah, thanks, Lily. That's I think a question that a lot of people wonder about is because the SDTM conversion process is a very analysis-like process, and historically FDA reviewers have been very distrustful of analyses coming from sponsors. It makes me wonder sometimes whether they should be equally distrustful of SDTM. So it's a good question. Thank you.
Right. And just to say, it's not distrust as much as it is a desire to ensure that we're all looking at the data in a way that answers our questions. And I think that sometimes the way that sponsors represent their analyses, they answer questions and they're presenting the analysis in a way that answers those questions. But as you're looking at it, as a reviewer, you may have other questions that you want to ask. And this is why we do it, right? This is why we do review. It's to make sure that we are considering other pieces. And it's not distrust as much as it is a desire to make sure that we are looking really thoroughly at whether there's impact of what we're seeing in front of us.
Good answer. Okay, so are we done? Are we done? Spit roasting Lilliam? Can we give her one more?
It's not a question. This is just an announcement that Lilliam will be at PharmaStat this year, so if anybody's going to that meeting, you can go ask her.
Lillian, thank you so much. Okay, so we're at break time. It's 10 after 3, West Coast time, and we're going to take a 20-minute break. While we're on the break, the folks on the webinar, it'll just be room noise or silent. At 3:30, I'll come back on and get everybody back in their seats, or try and get everybody back in their seats. And after we're done, if people want to stick around and chat, you can stay here till 6 o'clock if you want to. That's all good. And again, well, I'll do the thank yous after we get back together. We're on break now. Get people in from the hall. People in the hall need to come in. This thing. It's a pretty significant expense, and they were troopers about it, so please. I want to thank Patricia Gerend, who's our next speaker, and also David Tilio from TalentMine for having persuaded their management to underwrite this. Thank you. And thanks to Sandra Minjo for having talked them into it. Anyway, it takes the village. There's lots of people to thank for this one. This is all good. So our next presenter is Patricia Gerend. She's the one who's passing the mic around. Patty and I traded emails back and forth about how to introduce her, and we've both been around in the business for a while. And I was thinking, well, I could say that I've been in the business since presidents had beards. Well, it's not quite true, but actually, I think there is a metric that's meaningful, and that is we probably both started working on IBM mainframes, right? Yeah, been that long. and Patty's done a lot of things in those years, and actually, I'm not sure I'm even up to speed on what you're doing for Genentech right now, but you will tell us, right? And I just wanted to give a little bit of my own introduction. Not able to hear me as well. Let's see if that works any better. Okay. Testing 1, 2, 3. Yeah. I'll just point that away a little bit. The thing about site inspection data is that it's actually been a conditional requirement for some years, and this is data that FDA asks for to inform the Office of Scientific Investigations. Thank you. Inform them about where they should be looking if they're looking for issues in the chain of custody of data coming from the sites, and it's extremely useful to them. And Patty's going to talk a lot more about this, but the thing about it is that it really is a requirement, and very few people actually know about it, and it often takes people by surprise at the end of the process, where they have to put together information from people who they don't even know in their organization oftentimes, right? There's information that comes from clinical and ClinOps and regulatory and so forth and so on, but the main body of the data is often being put together by stat programmers. So it's really kind of problematic, and we frequently find that if companies leave it as a footnote at the end of their process, that this can actually end up being the thing that stands on their air hose and delays their submission. So anyway, this is an important topic, and one I think that a lot of people aren't that informed about. So anyway, with no further ado, let me give you Patricia Gerend.
Not having quite the height of a professional basketball player, if I stand behind here, I think you can barely see me. So I'll be walking around with my karaoke mic a little bit. The first thing I want to do is thank John, PharmaStat and his company, Ronnie, David, everybody. This was a somewhat challenging technical feat to pull off, and it has gone swimmingly, so good job. Okay, so as John said, we'll be talking about the inspection site selection data standards. That's a mouthful, probably mess it up at some point, but I'll do my best. The subjects we're going to cover, first of all, an introduction to FDA PhUSE working groups. And then we will talk about inspection site selection processes, the specific inspection site selection working group itself, the data elements within the site data set and how they relate to CDISC or don't, as the case may be. And then we'll go over what the next steps will be for this working group. And in addition to questions, I'm going to be asking people for suggestions. I know, we've had some really good questions and ideas come today already. Okay, FDA PhUSE working groups. This, I believe this was introduced earlier, the idea of what PhUSE is all about. I took this directly from their website because they said it better than anybody else could. The goal is to increase the efficiency of health product development and regulatory review. And the way they do that is they have the conference every year in March. It's the Computational Science Symposium. They also have working groups that get together at that conference but then carry on throughout the year. And they try to tackle significant issues in our business. The participants really run the gamut of the business. We've got FDA, industry, vendors, academia, did I miss anybody, CDISC. So it's really all comers. And for anybody who wants more information, there's a website you can go to. A little bit more on these FDA PhUSE working groups. They're all, I wouldn't say regulated, but coordinated by the PhUSE Steering Committee, and you'll see that in the center of the slide here. And there are currently 5 of them. At the top you see optimizing the use of data standards. And this group covers things like the analysis data reviewers guide, best practices for the use of standardized data. Traceability is going on right now, traceability for add-on and integrated data sets. Then we have emerging trends and technologies. This covers a lot of cool stuff. Think about the cloud, personal fitness monitors, how we can use that data, how we can use big data, things like that. There's a group looking at standard scripts for analysis and reporting, and they're using a variety of tools like SAS, R, S+, and looking at standard scripts for creating derived variables and reports, which is kind of a cool idea because when you think about it, every company, sometimes every project, even every study, reinvents the wheel. We're looking at rheumatoid arthritis endpoints and we're all deriving, tender, swollen joint counts, blah, blah, blah. Why not make the standard? That way everybody's on the same page, speaking the same language. We have the non-clinical roadmap. They're involved with SEN, which is Standard Exchange of Non-Clinical Data, which is, of course, for non-clinical data. And then we have semantic technology, and they're looking to use that technology for the entire life cycle of the study, really from protocol, case report forms, through data, through results. So there's a lot going on. But now let's jump to the topic at hand, which is inspection site selection. In the past, I don't know, like John said, we've been at this business a long time. In the past maybe 10 to 20 years, there's really been a huge increase in both the size and complexity of clinical trials. And with this has come specific challenges for the FDA selecting sites to inspect, such as there are more sites per trial, we have a lot more sites in other countries. They have finite resources. I think all of us can relate to that issue. The PDUFA timelines are set, and so increased efficiency is really mandated by that. And there have been a lot of variations on how sites were selected for audit. So the FDA responded to these challenges by developing a risk-based site inspection selection tool. And this does increase standardization, of course, as a result of that increases efficiency. And this has been going on I'm not sure when they rolled out the tool. I think it's approximately 5 years ago. So around that time, that was really my first experience with being asked for an inspection site selection data set. And we got this request, I think it was just weeks before the submission date. And you could almost hear the screams all the way to the Capitol. People were like, you want what? When? And yeah, as John indicated, yeah, this was kind of a problem. I mean, this data set, like he said, it involves stuff that we typically don't even deal with. We don't even necessarily know who does deal with it. So it was a bit challenging. In the green, you'll see the FDA's perspectives. Okay, they've produced this cool new tool that uses this risk-based approach to selecting sites for audit. They want the data that feeds into that tool, and you can certainly understand that. So that's the good part. That's why that's in green. Now we go to the maybe not so good part, which is in red. Industry's perspective was kind of what I just said, that what? Why do we have to do this? Why are we doing this at the last minute? I'm going to miss my timeline. Because as we said, the data in this site data set that they want isn't stuff we typically use. It's not needed for proving efficacy or safety. It's not needed for any of the stuff most of us here do as clinical trial programmers. So the data is difficult to produce, and as you'll see later in the talk, there's really not a lot of CDISC standards that can be applied to it. Okay, I was going to ask you a question before I go to the next slide. Okay, so here's the situation. The FDA wants something out of our clinical trial data. We're not currently prepared to provide that very efficiently, so what do you think we do about that? The title of the presentation is going to give you a big hint. We form a PhUSE working group, of course, and that is exactly what we did. This group was actually formed about 3 years ago at the symposium in 2012. And the original remit of the group was really pretty grandiose. We were going to do this and that. And then we started running into a little bit of pushback. According to the PhUSE Steering Committee, these working groups aren't really supposed to be establishing policy. And we were getting dangerously close to that by talking about things like, well, when in the process of a submission will this data set be requested and things like that. So we were asked, okay, take that off the table. The remit ended up boiling down really to just doing a gap analysis of what variables the FDA wanted in this data set and where we could find them in the CDISC standards. So we did a draft of that. And then a few things happened. The team went on hiatus due to job changes of most of the people in leadership positions. So not a whole lot has been going on for probably 1 to 2 years now. The group, however, reformed in April. We took the opportunity of being at the CSS meeting and talked to the FDA and said, are you still interested? They're like, oh yeah. So we decided to put it back together. The PhUSE Steering Committee said, okay, fine, do it. So it is now back together as of this month. And I'm going to be leading the group. The FDA co-lead is Jean Melinda, who was on the original team. And the CDISC co-lead is Nate Fremark, who was also on the original team. So that should help. Our remit now is primarily to complete that gap analysis. We really did make a pretty good start on it. The CDISC standards for identifying the gaps. And then once we have done that, we will submit that analysis to the CDISC organization, and they'll do with it whatever they decide to do with it. We'll be available to help them with questions and suggestions and things. A couple of years ago, I actually did a draft entity relationship diagram for all this information, but we'll see what happens. So that's the remit now, and the expected timeline will be that we'll complete the project within a year. Okay, so while this working group was meeting and deliberating and everything, the FDA issued a draft guidance about the site data set with an accompanying document that listed very detailed specifications like variable names, labels, descriptions, types, lengths, code values, all that kind of stuff. So feel free to take a look at those if you have an interest in this topic, because this is still the current draft out there. It has not yet been revised or finalized. But just to summarize it all for you, a summary about documents. Science inspections are needed to safeguard patients. I think we can all get behind that. You know, a lot of us take over-the-counter drugs, prescription drugs. We want them to be safe. We want the data. You know, as the gentleman here was asking questions about the faithfulness of the SDTM data to the raw data. We want the data to be right. And we want these sites to be inspected and have some kind of assurance that they're actually presenting something that's a real picture. The site data set is needed to assist in selection of sites for inspection, and it is needed only for pivotal studies. It should be submitted before the filing if possible, else with the filing in Module 5. And we'll get to the details in a minute, but this data set includes regulatory information, site-level efficacy, site-level safety, investigator information, a handful of other things, all by clinical site and treatment arm. The overview of this data set is that there are 39 variables in it. At this moment, 6, which is about 15%, do have explicit CDISC standards, and 38, which is 85%, do not. But one thing to note is the SDTM trial summary data set, the variable TS PARM is extensible. Does have an extensible code list. So quite honestly, I think some of these things could be stuffed into that data set. This number just represents the things that are explicitly provided for right now by CDISC. I'm going to go through all 39 of the variables, but before everybody starts yawning, I want to quickly say that we're not going to go through all of them in depth. I'm just going to put them on the screen. Feel free to read them as I'm talking. And there are a handful that I want to call out to you because I think they're important to discuss. They're broken down by category, so keep in mind every set of variables I'm going to give you is actually part of the same data set, all 39 of them in total. These are the regulatory variables. You know, nothing real exciting here. Our sponsor name was already in the trial summary data set. Something you might not be used to seeing, sponsor count. That's a count of how many different sponsor companies ran this trial. Because as you know, companies merge, they go belly up, they do whatever. The next group of variables are the study variables. And some of these are already in the TS domain. I think primary endpoint and endpoint type can go in there as well. Primary endpoint would be something like average decrease in blood pressure, let's say. Endpoint type would be something like continuous, discrete, time to event, that kind of thing. This slide represents these site variables, and these are the ones I want to talk about a little bit more. Let's take a look at what some of these are. Number of subjects screened versus number in the safety population, number discontinued, number of deaths, number of serious events, number of protocol violations, and then you see treatment efficacy, the standard deviation, all that kind of thing. So you can take a look at this list and realize almost immediately why this is important when the FDA goes to select sites for inspection. For example, let's say a site screened 100 patients and enrolled 5. Wouldn't you wonder why? Let's say your average efficacy across the study is 50%. You see one site with 100%. Again, you'd be raising your eyebrow wondering why. If you see a site with a lot of protocol violations, you might question all the data from that site. If you see an average discontinuation rate across the study of, say, 20%, the site has 90%, you're going to be asking questions. So it's really immediately obvious by looking at these variables why this is being requested. And these are the investigator variables, nothing too exciting here. Basically, what's my phone number, what's my email address. Until you get to the bottom, take a look at these. Financial disclosure amount, maximum financial disclosure amount. As John indicated in the introduction, does anybody here even have a clue where they could find this information? Certainly not on the case report form, certainly not on SDTM or Adam. And there are some people on the original working group who are even questioning whether or not information like that belongs in CDISC data sets. And yeah, I can understand the question. So to summarize the data situation that we're in here with the site-level data set, there are lots of data points needed that are not routinely produced by sponsors or used by sponsors. And as we said, we're not even sure where to find some of this stuff. Most of these do not have accompanying CDISC standards, which, I mean, that's what this meeting is all about. That's what CDISC has been about for how many years? 20 years. Standards really do increase efficiency, but we don't have them for this stuff. The data format, all of you in the audience who are astute database designers will recognize that was not a third normal form data set that I just showed you. So the data format is a bit challenging and complex. So our next step will be, our next steps, plural, the working group will complete that gap analysis that we talked about between what OSI wants to see and their site data set and the standards that CDISC currently accommodates. We're going to be working with the FDA on revising their draft guidance. And just a heads up to people, I think you may have caught this in Lilliam Rosario's talk, but she's talking about requiring actual treatment arm. I think for a while that was considered sort of optional. It either wasn't populated by sponsor companies, it was populated in a pretty inconsistent way. I mean, I can remember years ago going to the EX domain, just concatenating every unique treatment name and sticking that into ActArm. I don't think that's really what they want. So as industry people, we're going to have to get our acts together a bit on the concept of the actual treatment arm. So that's one thing that may be coming. And we'll be working with them, they'll be working with us towards the next revision of those documents. Then, of course, when we're finished with the gap analysis, we will submit it to the CDISC organization, and they'll deliberate whether or not there should be site-level SDTM information, site-level Adam information, what have you, and they will also, of course, be looking into appropriate data structures for that. And what the FDA has told us quite recently, this month in fact, is that if CDISC does decide to incorporate this stuff into their standards, then the FDA will be expecting us to provide the corresponding CDISC data sets to them, not necessarily this 39 variable data set. So, I mean, we're talking some time from now, but that's what we're aiming at. So as I said, I'd like to open it up not just to questions, but also suggestions. But first, I do want to acknowledge a few people who have made the restart of this effort possible. Michael Brennan was our original group, working group lead 3 years ago. Nate Freemark, who is now our CDISC co-lead, was also part of that group. Jean Melinda, who is our FDA co-lead, was also part of that group, as was Banu Khanna, also from the FDA. And it was really a conversation between Banu and myself at the recent CSS meeting that got this all going, so she deserves a lot of credit for that. Okay, so now to suggestions and questions, and some possible topics are, what do we think about putting site-level information into CDISC for this purpose? What do we think about putting in financial information? Do we have thoughts on anything else? So I'd like to open it up to the audience for questions. You know, I can probably do this if you want to. Hello?
I'm sorry.
Our database designer has a question.
I'm just going to have to be annoying. No, but what I'm kind of curious about is, have you had any discussions with the FDA in their tools that they're going to be using? Is it just going to be they're using SAS transport data set, or are they looking to, much like clinicaltrials.gov, where they use a backbone of XML in order to have the information? Because it's those discussions that really need to be, because, you know, if we're going to design tools for them, okay, I mean, they're designing their own tools, but I'm sure they're going to have some sort of a backbone for their tools that we can make our tools or our data more useful for their tools. They don't have to design it, we design it. Simple.
Yeah, that's a good question. Given that they're looking to, can people hear me?
Yeah.
Okay, given that they're looking to CDISC for solutions, whatever the data ends up looking like will follow whatever CDISC is doing, which at the moment is SAS transport files, but will very likely be moving on to something else in the future. So this in all likelihood will not be a unique circumstance of data submission. It will be the same as whatever CDISC is doing.
Because one other question, is this going to be, in other words, is the intent that basically an inspector is going to be able to take this information, okay, which includes, I'm assuming, all this site information, and they're going to choose these sites to review, and then they're going to have to, are they going to extract the data, or are we supplying the data to the inspector to go to the site and to review the source documents for the inspection?
That's a good question. I don't really know the answer to that. I'm not terribly involved in inspections. My guess is that they will use this information to pick the sites that look unusual in their eye, like the examples that I gave before. And I think what they do when they go there, they look at the actual patient records. I think they interview people. They compare them. And this gets to your earlier question at one of the prior talks about how do we know that the real data ended up in SPTM because we're doing all these transformations. They look at that kind of stuff when they do their inspections. So I hope that helps. Okay, do we have questions online?
No. Not yet. Hi, this is Hong from Clinton Insight. So I know right now sponsors are preparing the CSF for FDA reviewer to pick the site. Does the working group know of any sponsor who is looking at information and use them to do risk-based centralized monitoring during the conduct of the trial?
Actually, that's a really good question. And I mean, as I said, the working group is just restarting now, but back when we were in full swing 2 or 3 years ago, there was a lot of talk about sponsor companies finding out what did the FDA care about, what did they look at, what was the trigger in their mind for somebody to be selected as a site for audit. So yeah, absolutely. I think by looking at what the FDA is looking at, we could certainly improve the way that we audit our own sites, absolutely.
Patty, I'd like to suggest that you expand your scope. I think the question of whether or not these variables are found in CDISC is too limited, because a number of the variables that you listed, I'm fairly sure, are already in the e-submission XML backbone from regulatory. A number of the variables, I think, could be summarized from the data that's already in CDISC, such as the number of adverse events or serious adverse events by site. So I'd like to suggest that you perhaps expand the scope to not only is it within CDISC, but is it anywhere within the submission already? Because FDA doesn't like us to submit something twice. We don't like to submit something twice or more than twice because we have to QC it against each other instead of that. So just the scope of whether or not it's within CDISC, I think, is a little limiting. So I'd like to suggest that it's expanded a little bit on that.
That's a good comment, and that's something that came up in the original working group. there were industry representatives that said, why are we even looking at that? Because as you just indicated, most or all of this information is already submitted somewhere. It might be submitted in a textual paragraph someplace. The financial disclosure stuff, I'm sure, is submitted somewhere, maybe in a table, certainly not in a CES transport file. So you're right that the majority of this already is. And it could be possible that there is a way to pull it out of the background. I guess that's something we probably should look into. But, you know, at a minimum, we will do our gap analysis. We'll talk about other approaches because some of the people that have re-upped to be on the working group are the ones who are saying what you just said, that, hey, we give this to you guys already. Why do we have to do it again? Why don't you figure out where it is, and you pull it out, and you make this data set? So we'll see. Good point.
So I have a couple of comments. We just did a submission, and we create inside XPT. So actually, we use the SDTM, and also some information from clinical operation, like the financial disclosure, and also we create XPT file, and our vendor create the define. So actually, it's a different section in the submission. So your clinical database, clinical submission is in one folder, right? This is a totally separate folder. It's not your clinical data, not for the review. This is for audit only. So we use two data sources from SDTM and clinical operation data, and also we also use that data to do some mock audit. We also select some site to do audit. Yeah. So this I consider this like item based on SDTM.
Okay, thank you.
I had one question about one of your standard variables that I think I understand why you deleted over and you didn't discuss it. That's the inclusion of the domain variable. What was the thinking behind originally including that in the standard?
I can't exactly answer that question, but I do know what value the FDA is anticipating. We put in there the value DE, which stands for data elements. And my understanding is that comes from the fact that that value was arrived at by the tool developers. These are not people that are analyzing SDTM or ATOM data. They're just doing, you know, as you just said, they're just doing this one task. And so to them, oh, these are the data elements. Well, to us, these are 39 of a huge number of data elements. So what they're looking for is a domain name for this 39 variable data set. I'm just not sure that's how it's going to shake out. And if there is one, because this is really not it's not an SDTM structured data set for sure. If CDISC does, you know, decide to go with this, we will be looking at several data sets. Some may be SDTM level, some may be ATOM, and there likely won't be just one new domain name if we even go that route, as we talked about. But that's what their thinking was. DE is the domain name for data elements, and that represents the 39 variable site data set.
Hi, Patty, it's Linda. So I'm thinking as I'm looking at this that yes, you know, it is possible to do a lot of these summaries at the site level and have it be part of the tables that you produce as part of your submission. And I can see why you might want to do this because that's liable to be scattered all over the place. When I was at the PhUSE meeting, one of the working groups there is working on ways of storing analysis result information itself in a machine-readable form that could eventually go into a submission. It looks as though that effort's got, you know, some ways to go, but I wonder if anybody in your group is in communication with that group about whether this level of summary is something that could be eventually rolled into results to be submitted.
Are you talking about the semantic technology group?
Yeah.
I think that's worth considering. You know, right now our group has just reformed. Initially, we were not looking into that, but since before the law has happened. So yeah, I think we'll add that to the list of, like looking at the XML background as an idea, semantic technology as an idea, CDISC as an idea. At the moment, we are focused on CDISC, but I think it sure doesn't hurt to look at other potential approaches. Yeah, thanks.
Okay, the question from online is, does this information belong to SDTM or ATOM? My understanding is that SDTM should just capture raw data and contains very little derived variables. We're already deriving some of these variables in the ATOM datasets.
Yeah, as I was saying a minute ago, this 39 variable data set is a mishmash in my mind of things that could be specified in SDTM and things that could be summarized in ATOM data sets, like the average efficacy per site, the number of serious adverse events per site. Yeah, those are pretty clearly analysis measurements, so that does sound like ATOM to me as well.
More questions?
Thank you, Patty. Well, thanks, everybody.
Before we move on to the last event, actually I was going to ask Dave Warles to say a little something. He came out to me at the beginning of the break and was telling me that actually there is a way, apparently, for sponsors to get access to JumpStart results.
Hi. So this question came up at our company, and we learned from Pinnacle 21. They are the for-profit arm of the Open CDISC team. They created Open CDISC Enterprise, which is the application that the FDA is using to do the JumpStart process. They also will, for a fee, do that same service for any sponsor company. It's based on a per study flat fee cost, and they perform the same. They are the ones that actually do the JumpStart analysis at the FDA, and they can do the same thing for you. So you would have to contact Pinnacle 21, and they would be able to set that up for you so you would know ahead of time what your overall numeric score is and any of the issues and findings that they have. They use the same application, Open CDISC Enterprise, so it is possible for you to get access to that information and know what it's going to be before you ship your submission off to the FDA.
Okay, so our last bit of business is actually just it's sort of a round table about the CSS PhUSE conference, which we've heard from various people about in little ways today. So let me get something up on the screen for you here. And Mickey, can you verify that we are live with them one more time? Okay. So we're live with that on the screen. Okay, good. For people who have not, actually, let me first see a show of hands. Who went to the one this year in March? Yeah, 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12. That's pretty good. I think I did a count a couple of days ago. There were 20 people from the Bay Area. The total attendance at that conference was about 256 or so. Were they hearing Patty okay? Okay, let me just get closer then. From my point of view, this is one of the most important conferences of the year for people in biometrics. And the reason is really, really simple. You go there, and the mission at that conference is not to sit around and listen to presentations, but rather to roll up your sleeves and get some work done with FDA people. And so there's a bunch of FDA people there. There were more than 50 signed up at this year's conference, and they just circulate in the room with everybody else, and it's the most wonderful opportunity to just sit down, standing at the where the cookies are, or just out in the hall milling or wherever, and strike up a casual conversation with people at FDA who know things that you need to know. How your data are being used when they show up, on the FDA's doorstep, and what their expectations are, and they have that intel for you, and they're looking for the same kind of intel from you, from industry. One of the reasons they love to go to this conference is because they get lots of information from the industry people who go there and who they work with about what kind of churn is involved, for example, in getting good data to the FDA, and what the operational implications are of a new standard or whatever. So it's important to them, and it's important to us, and I would really, really recommend that that next year when this comes up, that we send lots more people from the Bay Area to it. It's really important. So what I've asked is for a few people who were there, who I saw were there, to actually come up and give their impressions and talk about some of the things that are under discussion and projects that are being worked on by these working groups. So Sandra, you're one. Amy, Pan Yang, Lisa Brooks, and Wei. Is Wei still here? And did I ask anyone else? Oh, Linda. Yeah, Linda, you're up. And Mickey, too.
Mickey's busy back there.
Okay. So this is not the entire list. The stuff in blue. Is those are the big working groups. The things in black are.
We only have 4 of them.
I know. This is not all. This is not the whole list. The things in black are the projects that those working groups are working on, and this is the rest of the list. So the list that's on the screen right now, probably 60% of all of the people who went to the conference were involved in these activities, and the other 40% were involved in all the rest of these activities put together. So there's some interesting stuff going on, and there's some stuff that really is to be watched, because it could turn into something pretty interesting and useful. But it's got a longer horizon than some of the than, for instance, the study data standardization plan. So let me ask, first of all, let's see, we have 2 microphones. We have 3 with the podium. Why don't we start at this end and just have everybody introduce themselves, name, where they're from, and what stuff that they were involved in, or what they sort of.
So my name is Wang Meng, and I'm from Genentech Roche. And this is the second time that I attend the PhUSE conference. And I think one of the key takeaways that I did is the first time I tried to explore everything, all the working groups. And that's a bit of a challenge because so the second time what I did is I actually just choose a few that I go. And the one that I end up with is the ODS, the optimization data standards. And I find that by just kind of follow the same group, you get to know the people and you interact with them. And right now, I'm actually actively participating in the best practices, the subgroup within that ODS group.
My name is Wei Zhong from JumpStart Science. I went to this year's conference. Actually, that was my first time attending PhUSE. I heard of it before. And what I found I like this conference, as John mentioned earlier, it's very different from the regular conference because in the conference you really work on issues, roll your sleeves. That's very different. The other thing I like this conference is there are a lot more FDA people, FDA reviewers coming, and also as you might have heard, the first two presentations today, and also I think I find it's very important for me to really learn from, always interesting to learn from FDA reviews point of view, how do they look at data, and I think that also helps me to prepare our submission data and do our study work. And for me, I think there are altogether 5 work streams. I participated in one because it's hard for me to participate more than one. I went to the optimizing the use of data standard, and also there are several topics within the work stream. As a particular, I think I end up with the same work group with the firm, which is try to come out with a best practice between defined.xml and also the data guide for reviewers guide. And there's a working group. I think we meet every 2 weeks. And also, I think there are there are over 20 issues we list and we're going to work on. I think the goal for this group is by the end of this year, we'll come up with a white paper as a recommendation.
Right, yeah, and let me add to that. One of the very cool things is that the FDA folks who show up, they're not just the suits. Right? These are folks who actually do clinical reviews and are trying to use the stuff that Lillian was talking about in her presentation.
Yeah, to give a plug, the 2 first presenters we had today, we got by being at this meeting. John met them, asked them if they'd be willing to do this via a web, and they were really open to that. So it would have been really hard to get these guys to try to do this via email. But being in the same room with them, they were both totally open to it. So it's a really nice way to meet a lot of different people. And you don't know as you walk around. You don't know who these people are. They're just mixed in with everybody else. You don't realize you're having lunch with an FDA reviewer unless you read the fine print on their badge. So it's kind of neat. So I am one of the co-leads on the traceability and data flow under the optimizing the use of data standards. Somebody phrased that as ODS just before the meeting, and I tried to cut it immediately. We're not going to call it ODS. OUDS I'm fine with, but it will not be ODS. So I'm working on the analysis data process flow. So I'm on the ADAM team also, and I'm trying to provide sort of a link between what the ADAM group is doing, which is really just around structure, variable names, that sort of thing. And there's a gap out there in process and data flow. So how can we implement the ADAM standards into our data flow? And how do we want to make this happen? And it's going to cover things like, do you need an analysis data set before ADSL?
because you're doing a lot of work to create variables in ADSL, which is only one record per subject, and there's not a lot of traceability there. Do you need an interim or intermediate data set on the way to producing a very complex analysis data set? So that's what that group is doing. I also sat in on the other traceability and data flow, which is the integration. Again, very much working in cohort with the ADAM team on integration, same issues. ADAM team is working on structure and data set variables, that sort of thing. This group is working on, well, there's so many different ways to put an ISS together. What are the best practices for your way to do it? You know, are you going to combine SDTM and make an SDTM integration and then use that to drive ADAM? Are you going to combine your atoms together? Are you going to do some studies one way and some studies another? You know, are you doing one way for safety and one way for efficacy for an ISS versus an ISE? So there's a lot of different options out there, and this group is trying to get a handle on all those options and come up with some best practices for that. I also helped lead one of the roundtable discussions on the FDA guidance documents. So trying to come up with what can we as an industry do to help standardize some of the things that FDA is looking for, so that it can become more of a filling out a template rather than having each of us create something different from scratch. And that's how the ADRG and SDRG came about. FDA said they wanted these documents. The CSS working group said, okay, what should they look like? And came up with these templates. So that's really what that group's trying to do. I think that's it. It's involved in all that.
My name is Professor Martin to the inside. So this year is my third year attending the FDA PhUSE CSS conference. I really like that conference. If I don't have budget to go to one conference per year, the conference I will sign up. But the key benefit is I get to network with the FDA, clinical reviewer, and statistical reviewer. It's a very friendly setting. I ask them questions that puzzle. For example, as the statistical reviewers, what is their expectation on submitting SAS programs? How would they use this? How would they review those programs? And I ask several of them. So I feel if I get an answer from them directly, I'm in a better position to advise my clients, you know, when it comes to the time to prepare the submission SAS programs. And then the working group is also very helpful. I participated in the SP group. And then the benefit of this conference, people are very open to share their outputs. Everything is published, unlike the DIA conference where they never publish anything. This one, all the keynote speech presentations, FDA posters, and a working group white papers are published on the PhUSE website. So I encourage the people who didn't get to go, they can go to the website and read these publications. They're very helpful to my daily work.
Hi, I'm Amy Flotman. I work at Genentech. And I was in the semantic technology team of PCSS for 2 years running. I'm specifically, it's not listed up there, on the representing CDISC vertical representation model on RDF. And that's what it's used.
Yeah, I sort of rolled that up into the foundational standards for RDF.
Yeah, it's not a big project. We started small. That's really smart people would do. We started with a schedule of activities that's currently in beta testing on GitHub. You can find it on the PhUSE website. Now we're working on eligibility modeling, trial registration, outcomes and objectives, and there'll be more after that, of course. And our leads do work with the Trans-Already Biopharma Common Protocol Template Tech team to make sure we can link to any work that they're doing. We don't duplicate efforts, we aren't making false assumptions, things like that. So we definitely try to reach out to people that are working on similar things, just taking from a different perspective. And Patty, I'll be happy to bring up the link into the work that you're doing so that we can all work together. At least be aware of where the touch points are.
So the semantic technology one, most of the stuff that they're doing in that is modeling in RDF.
Correct.
And RDF is a very fast up and coming modeling language, and somewhere along the line we're all going to need to be dealing with it. So this is not a this month next 6 months kind of time frame for most of the stuff that's happening in there that we'll all be dealing with it. But I'm pretty sure that over the next couple of years, this is going to creep into our everyday lives.
We're really excited about that XML representation of data, finally real machine readable.
Right.
Yeah, one quick question. You guys better be very, very aware and careful with XML data. Take a lab data set and try to convert it to XML. You will crash any system you put it on. Normally, when you create it to be size, most people don't know that is normally the maximum size of any file for the Windows operating system. It's very, very quirky on Linux and any other, and try to run it through a browser, and it will definitely crash. Okay, so just be aware. XML is fine, but it is text-based.
But not correct.
And can create, I've done it before. I've taken a 300 megabyte laboratory data set, converted it to XML. It well exceeds over 200 gigabytes, and your lab data will become garbage.
200
Just keep yourself aware of the tech behind this, okay?
Wow.
Well, that's a really interesting point. I remember turning Firewall a few years ago when everybody was getting involved in defining XML. I used to do presentations where I'd say, all right, so how many people in this room can spell XML? and no hands would go up. And I kind of feel a little bit in that boat because before this meeting I probably couldn't have spelled RDF. I've never heard of it before. And I wound up going to some of the meetings on modeling analysis results and metadata in RDF. And mainly because it was the closest thing that I could see to analysis results metadata, which I'm quite interested in. We're starting to implement that in Define 2.0 because now you can. And that group is actually not doing that per se. They're doing something a little bit different. They are working on developing RDF models for the results themselves, the actual numerical results, and how you represent the context around it. Which is why I was asking Patty that question, because theoretically, if you're looking at things like site-specific efficacy results and their standard deviations, it could theoretically be modeled in that. It's going to be a little while before that group actually has enough traction to produce something that people can actually use and submit. But it's a really, really interesting thing to keep track of. And it also made me aware of the fact, which I hadn't been particularly conscious of before, that the CDISC organization itself is using RDF to represent the standards, which is another good reason to be aware of RDF and to start getting a grip on it. So both of those things, I think, were to me really kind of eye-opening and, sort of make me aware that this is something to really watch because there are going to be some very interesting things happening along these lines.
Hello, I'm Lisa Brooks, and I don't get to go to conferences very often, but the last few times I've chosen to go to a conference, it was this one, and the reason why is because I love data standards, and I love talking about them with people like you. So I was involved in the Study Data Standardization Plan group, and I'm actually the lead for that SDSP example. I chose that group mainly because I help pharmaceutical companies do their data submissions, and I try to get involved as early as possible. And I found in the last few new drug applications that I've done that our sponsors even breach the subject, and so I'm very, very interested in this topic. So that's what I've been doing. And we had a meeting this morning, which was very exciting. We plan to have a template out by the end of June, But I heard one Fitzmartin say something today, and you can tell me if you heard him say that too. He said that he was going to have a guidance published on the content of the SDSP. And I want to know, are we going to have that guidance before we have a template?
That's not what I heard. What I heard, and I may have heard wrong, what I heard was that the current guidance actually has a couple of paragraphs about the need for a standardization plan, and so it's mentioned in guidance already. It's mentioned in guidance, but I didn't hear him say that they were going to come out with a guidance about it.
I don't know if he used the word guidance. I think that's the wrong word. He said that they were going to provide additional information. A recommendation. So I hope we get that recommendation soon.
Right. Yeah. Right. Okay. So Lisa has actually gone to the effort of putting together a set of slides about the SDSP, and I think it's a really good idea to give this a little more airtime because like the study data reviewer's guide and the analysis data reviewer's guide, this is something that will eventually be mentioned, be pointed to in guidance and technical specifications in FDA documents. And it's, from my point of view, it's one of the most important deliverables that's going to come out of the PhUSE effort this year. So thank you all for doing that, and I'm going to turn over the stage to Lisa now.
Okay. I'll just use this one here. I'm going to make this brief. You all have been here for a long time, so I'll kind of. What I really want to do right now is give you a peek at what the SDSP is going to look like. It's up on the PhUSE website, but it's not open for comments yet. So we just got a bunch of comments today, so people are very excited about it. The team kicked off in July 2014. We met at the CSS meeting last month, and we're meeting twice a month via teleconference. And we met this morning, like I said. Our goal is to provide a template, like instructions for how to create the document, as well as an example. It will include study information for both clinical and non-clinical data, and we are collaborating with our non-clinical partners on other PhUSE working groups. And like I said, we want to have a template at the end of the month, at the end of June, and hopefully in the fall we'll have the instructions and the example. So what is the SDSP and why do we need it? In the study data technical conformance guide that we heard a lot about today, I placed this excerpt where at the pre-IND stage or the IND stage and then throughout the development of your study product, FDA wants to know what you have planned. And these are the types of things they'd like to have included in the study guide. What is the list of planned studies, the types of studies, what design you're using, and what data standards you're using. And that, the list of data standards, of course, are in the catalog, so you can just reference that. Let's go to the next slide. So it's a living document. Like I said, you'll be submitting it, starting at the pre-IND, IND stage, and then, throughout, up until you have your NDA plan or your BLA. And it should be periodically sent. Now, the FDA has warned us. They don't want it every time you add a new study. There's key time points, and I think that that's a mystery, and I think I need to ask that question. But this plan is supposed to be, and it's always in debate, you have one plan for each compound and for each indication of that compound. So you might have to copy this over if you're submitting your licensing application for multiple indications. Okay, so I've just kind of thrown up here on the screen, some excerpts from the actual template that we're working on. This is very draft. Like I said, we're going to get some more feedback from FDA, and we haven't gone through our comment period yet. But this is just a list of the non-clinical studies and the clinical studies, and the types of information we're going to include. I'm not going to go through all of it. And this is where we're listing our standard and control terminologies. It's just by study. Are you using, which version of SDTM-IG are you using? What version of Medra? Any other standards that you're using? Like I can imagine you might need a TALOG up there, one of the therapeutic area guides. And I think I'm going to be using this one a lot. This one is when you don't use a standard, you use something that is not expected, and why you want to use that. Now, I work on a lot of NDAs that have very old legacy data coming from previous sponsors, and new sponsors have licensed it, and a lot of the data really isn't applicable anymore to the submission, but you need to include it, and this might be where some of those studies might have done. Okay, and this is probably going to be completed near the end of your product development, where you talk about what sort of pooled analysis you're going to do, safety, efficacy, and what standards you're going to use for that. And the last page is just a list of the studies in which pooled calculations they're going into. And I've just included the references here with links to the different information, also a link to the PhUSE wiki so that you can see the template for yourself and the progress that we're making. And that's my email address up there if you are interested in asking questions or would like to know about any future.
We're going to be posting the presentations, assuming I can get them from Lilliam and Ron. We're going to be posting all the presentations on the CDISC portal for this group, and we've also been recording the webinar, and we're going to try and post that recording as well, so that you can replay for people back at work if you didn't see it, and so forth. So
thank you guys.
I think that's it for today. Thank you for coming.
I want to give a shout out before you guys leave. John from PharmaStat. In the back of the room, we have Nikki also from PharmaStat. Dave from PharmaStat set up the webinar. They paid the difference in price between what our two official sponsors did and what this cost. So big shout out to PharmaStat and